Hi and hello everyone. I am Parthasarathi. Welcome you all for today's session. 21st century education won't be defined by any new technology. It won't be defined by one-to-one technology programs or tech-intensive projects. 21st century education will, however, be defined by a fundamental shift in what we are teaching, a shift towards learner-centered education and creating creative thinkers, says Carl Fish. Today's webinar on designer crops is brought to you by the Biomics team. The speaker of today's webinar is an eminent resource person, Dr. M. Muthamilarasan, Assistant Professor in the Department of Plant Sciences from the University of Hyderabad. He will be imparting advanced knowledge on the designer crops. It's pride and privilege for our Biomics team in hosting this webinar with an eminent speaker. This webinar enriches you with the correct knowledge of next generation genomics tools for crop improvement, gene and genome editing approaches, and issues associated with genetic engineering and biotechnology. Welcome the challengers. Look for the opportunities in every situation to learn and grow in wisdom, says Brain Tracy. I am very glad that Biomics steps up in new heights in organizing a series of webinar talks. We believe in mutual growth and development. On envisioning our motto, this webinar talks are the new initiatives on our Biomics platform. I would like to welcome our eminent resource person, Dr. M. Muthamilarasan, Ph.D., Assistant Professor from University of Hyderabad, on behalf of the Biomics team. Welcome you, sir. Our speaker is an eminent scholar in the area of plant sciences. Henceforth, I believe that this webinar could be nourishing the young minds in the field of crop biotechnology. Now I welcome dear Sachin, research scholar, Indian Institute of Science, IISc, Bangalore, and Vishal Kumar Chagu, who are admins of the Biomics. Cordial welcome to you, both of you, dears. And I also appreciate and welcome all the scientists, deans, professors, and students from various colleges, universities, and research institutes globally who joined with us through this webinar. Our eminent resource person, Dr. Muthamilarasan, has earned his post-graduation from Madurai Kamaraj University and got his PhD from National Institute of Plant Genome Research, New Delhi. He is currently working on the molecular genetics and genomics of chosen millet species. Throughout his scientific career, Dr. Muthamilarasan has contributed significantly to the structural and functional genomics studies of the model crops, foxtail millet. In structural genomics aspect, he has developed several large-scale genome-wide molecular markers to understand their associations with key agronomic and climate-resilient traits. He has further demonstrated the use of these markers in genomic assisted breeding of Gramineaceae species. In the area of functional genomics, he has characterized prominent stress-responsive gene families and investigated their expression patterns in response of various abiotic stresses. The genetic and genomic resources developed by him have also been made available in comprehensive open access databases for expediting structural, functional, and comprehensive genomic research in millets, cereals, and bioenergy grasses. Dr. Muthamilarasan is the recipient of several awards, and recent ones are Fulbright Nehru Alumni Award 2019, INSA Medal for Young Scientists 2018, Nasi Young Scientist Platinum Jubilee Award in 2018, Early Career Research Award 2018. Now, I would request our respected professor to make more insights on designer crops so that the enhanced understanding could help us better to serve our country and its people through research and development in the field of plant sciences. Before that, I request the audience, if you have any questions during presentation, please start typing them in the text box. Our team will do their best to convey your questions to the guest speaker. Sir, please.
Thank you, Partha, and thanks to the team Biomics. It's a nice opportunity to meet you all people. So, without wasting any time, let me go into my presentation. I'm just stopping my video to reduce the load on the internet actually, so that my voice would be clear. Yeah, is my screen visible?
Yes, sir. Okay.
Okay. Yeah. Again, good morning, everyone. It's nice meeting you all through this virtual platform. Actually, though we are in a different phase of facing the ongoing pandemic, I could see that I just went to this YouTube portal where it is getting live relayed, and I saw that students are actively participating in this live stream of this talk in large number, of course. So this clearly shows that nothing can stop us from learning. And thanks to Partha, as I said before, and the team Biomics also for hosting this program online. So yeah, today I'm going to talk on designer crops. So we will go step by step. I mean, I'll be having few slides which you may find elementary, but it will be useful for undergraduate or postgraduate students who can understand the basics first before getting into the actual thing which is being carried out in the national laboratories or international labs. So getting into this, I begin my talk by dedicating this to Tiruvallur, who regarded agriculture as the foremost profession in the world. This Tirukkural, it says that agriculture is the foremost profession in the world, and when we talk about the world in which we live in, let's not forget that we are nothing when compared to the size of our galaxy, and we are not the permanent residents of this earth. By keeping this in mind, and having said this, let's also not forget that we are the last ones to evolve on this earth. If you could see the evolutionary trajectory, our ancestor Homo erectus, it was just 1.8 million years ago, and our Homo sapiens, he evolved only 200,000 years ago. While other components, let it be biotic or abiotic factors of this earth, has evolved millions of years ago. Though we came at the last, we are leading the game in terms of number. If we could see today morning at around 5 o'clock, I was taking this screenshot, which shows that our current population is 7.8 billion. So it is still growing on. So in a way, though we came in the last, we are leading the game in terms of numbers. And experts predict that, or they project that by 2050 we will reach 10 billion. So as of now we are 7.8 billion. And since our kindergarten, we have been told that there are three basic needs for a human, namely food, shelter, and clothes. We have been told that without any one of these three basic needs, a human cannot survive. But the question is whether all the 7.8 billion have got these three basic needs. The answer is a big no. 1 in 10 of us, that is 8 to 1 million, are hungry. It means that 821 million people among us, they go to bed in the night with an empty stomach. For every 10 seconds, a child dies from hunger-related causes. And on the other hand, 1 in 4 children suffer from malnutrition, which shows that it is not just the food alone will serve the purpose, but getting the nutritious food will. So this is the scenario. And I hope everybody would have seen this picture of a famine-stricken girl who was later found to be a boy in Sudan. And this child is attempting to reach the United Nations feeding center, which is about half a mile away. And the vulture is waiting for the child to die so that it can feed the dead body. Kevin Carter, who has photographed this event, won the Pulitzer Prize in 1994. This picture was captured in 1993. So this is a scenario which has happened in 1993. Though the picture was captured more than 2 decades ago, nothing has changed much in terms of hunger. if you could see this global hunger index 2019, you can see that South Africa's or the Africas altogether and the South Asia are still in the danger zone of hunger. This yellow indicates the seriousness of the hunger in the regions. And the global hunger index scores of year 2000, 2005, 2010 and 2019 underlines that there is no significant improvement in the mortality rate of children below 5 years of age. This black one, there is no significant improvement in the wasting and stunting of children, and there is no significant improvement in the proportion of undernourished. 20 years ago, it was 38.4, now it is 29.3. So this ratio is coming down, but it is coming down very slowly. And this shows the progress of countries that have made an effort in reducing this level of hunger among its situation. If you could see, India is somewhere here, which is still in the serious zone. Only one thing is we are performing better than Afghanistan, but if you could compare us with our neighboring states, it shows that India is far behind them. Other neighboring states like Sri Lanka, Myanmar, Nepal, Bangladesh, Pakistan, they are performing better than us in terms of eradicating the hunger. So yeah, it apparently shows that our progress is evidently not up to the mark. Except Pakistan, all other neighbor countries are performing better. At this point of time, I would recommend you people to read the global report on food crisis for the year 2020, which is released by in collaboration with several international agencies like FAO, WHO, World Food Program, UNICEF, etc., etc. The report was released before the full impact of COVID-19 was realized. So this was released before the full impact of COVID-19 was realized. However, in such normal case itself, the report shows that 135 million people living in 55 countries are in acute food insecurity situations. Again, I am repeating that it is showing that 135 million people living in 55 different countries are facing acute food insecurity. This population is highly vulnerable since any event like the ongoing pandemic can easily cause famine and starvation. Now, what is happening is, since 2015, we have been investing so much effort in reducing the people who are living in extreme poverty. If you could see, millions of people, it was 740 million people in 2015, they were in poverty. The line was drastically going down, but an event like the ongoing pandemic, it just put everything back into how it was 5 years ago. It is almost like, the June downside projection is almost similar to what it was in 2015. So what are the effort we have invested in these 3 years has went back? Or even you can include these 2 years also. In 5 years, it has went back. So this is the effect of, so what it means that for people like us, we are facing only one crisis as of now, that is this COVID-19. But for predominant amount of people, they are facing two crises or dual crises. One is COVID-19 and the other one is poverty or hunger. So they are battling with these two. So, and this World Food Program projection, it also shows that the risk of hunger pandemics, as coronavirus said, to almost double. So it is saying that the 130 million people, which I have shown, 135 or 135 million people, which I have shown here, they are projecting that this would have been doubled by now because of this COVID-19. So that is what our newspaper is also saying, that global hunger double due to COVID-19 blow, says UN. So they are saying that these people don't need a disease. Even without disease, their lives were hanging by a thread. So that is the effect of, that is the impact of this food that is causing into the global, human race. So at this point of time, I would recommend you all to visit this website. This is an interactive website where you can see the real-time effect of hunger and malnutrition around the globe. So that is, for you to see, yeah. When coming back to the story, I want to show you a few statistics on the effect of food insecurity among children. The joint malnutrition estimates released by UNICEF, WHO, and World Bank Group, they show that 144 million children under 5 years of age are stunted, 47 million children under 5 years of age are too thin, 38 million children under 5 years of age are overweight. When you see the percentage of stunted children, South Asia again tops. It has more than 30% of stunted children, that accounts for 55.9 million in number. In case of wasted or in case of too thin for their age, again we top with 14.3%, which amounts to 25.2 million children. So, and since stunting and wasting are more in South Asia, obviously overweight children will be less. However, the northern and southern parts of Africa has an alarming percentage of overweight children. In Australia and New Zealand, the numbers are way too high, as you could see here. So, to build a better world for people of all levels by 2030, the United Nations has formulated 17 Sustainable Development Goals. You people might be well aware of these 17 goals. If you could see, among these 17, 3 are directly related to food. And this underlines the severity of the issue I'm talking about, and it is at the verge of getting worse due to the ongoing pandemic. So when we talk about food, many of us are ignorant of the fact that I'm going to show in my following slides. The largest and the most diverse botanical garden, that is this UK Kew Gardens in United Kingdom, has listed that there are a total of 11,28,000 vascular plants in this world. There are a total of 11,28,000 vascular plant species in this world. Among this 11 lakh something, archaeological and fossil evidences shows that approximately 7,000 were used as food by our early humans, which in due course of time got shrunken to just 3 crops, namely rice, wheat, and maize, that cater more than 50% of world's food. It is actually 11,28,000 vascular plants. Our early humans were consuming approximately 7,000 plants. Now we are having only 3 crops on our plate, which caters up to 50% of the world's food. In addition to these 3, there are 12 more crops or plants that altogether with 5 animal species supplement 75% of the world's food. So now you may understand the questions in reality, how you will feed 1.8 billion people with just 15 species in your hand. No matter how much effort you invest in improving rice, wheat, or maize, no matter how much effort you invest in improving other vegetables, let it be tomato or let it be brinjal, let it be any major crop which belongs to these 12 species, you cannot cater 7.8 billion people with just 15 plant species. So that is where we people are advocating for 2 important things. But one thing you have to remember is, as long as this discrepancy is not addressed, hunger and malnutrition issues cannot be solved. As long as this discrepancy is not addressed, famine and starvation could not be averted. As long as this discrepancy is not addressed, life and livelihood of all humans on this earth cannot be ensured. These 3 things you should remember, and that is where we plant biotechnologists advocate for 2 important points. Number 1 is mainstreaming underutilized crops for food and nutrition security, and using biotechnology to produce better varieties. That is what we call as designer crops. So I'll quickly spend little more time in telling you people what is this underutilized crops, and then we will come into our main topic of today's discussion, that is this, how to use biotechnology to produce varieties that can better withstand or that can easily cater or that can quickly cater the needs of the 7.8 billion. So whatever I'm going to talk today is an excerpt of my article published in 2018, that is on multi-omics approaches for strategic improvement of stress tolerance in underutilized crop species, published in Advances in Genetics. So first comes mainstreaming the neglected and underutilized crop species. What do we mean by this underutilized crops? That is the first thing we have to understand now. Except the 15 plant species that I have showed you a minute ago, rest all are neglected and underutilized. Why we call them neglected is they were totally ignored by the mainstream research. If you could see, research on rice, wheat and maize gets much attention as well as funding, but other crops receives less research attention. Among vegetables, tomato is in limelight of research. While talking about funding, I should also inform you that even private players are also interested, in working on rice, wheat or maize. For example, the Melinda Gates Foundation of the Microsoft founder Bill Gates gave $15 billion to International Rice Research Institute in Philippines to engineer C4 photosynthetic pathway in rice. In such limelights, our underutilized crops won't come. And coming to this underutilized part, these crops are cultivated only in certain regions and consumed by a particular group of population. So these underutilized crops can be tubers, legumes, leafy vegetables, and minor millets. I will show you a few examples of these. So as I said, these species are highly adapted to marginal environments. They can easily grow under low input conditions and stressful growing conditions. They are highly nutritious, such that they can contribute to healthier diets worldwide, and particularly for rural poor. They have potential for the development of novel consumer products to generate income for smallholder farmers and their communities. And these species are typically embedded within the local cultural traditions, such that greater use could also strengthen local identities and contribute to empower the marginalized communities. So in the next few slides, I will show you some examples of underutilized crops, and then I will quickly move to demonstrate how next-gen genomics tools helps in studying one such crop, or a few examples out of it. So these are some examples of underutilized tubers. You could see taro is there, arrowroot is there, Indian shoot is there, guanala, purple yam, air potato, elephant food. If you ask me whether one can survive only by eating such tuber, then the answer is yes. Predominant African community relies on cassava as their staple food. Even tubers are also a major constituent in the diets of Keralites, if you know. So tubers such of those tubers are still underutilized and neglected. These never came into the mainstream research, or these never came into the plates, or never came to address, though they have the potential to address food and nutrition security, they have not been mainstreamed yet. Coming to the legume part, there are several species that are not much prominent. For example, this winged bean, jack bean, Angora groundnut, kidney bean, faba bean, lima bean, cowpea, horse gram, rice bean, velvet bean, and atsiki beans. Coming to the millets, we know much about major millets like sorghum and pearl millet. The millets other than this sorghum and pearl millet constitute the small or minor millets. This includes finger millet, foxtail millet, proso millet, little millet, barnyard millet, teff, kodo, fonio, job steers, guinea, and brown top millets. I'll give little more information about these millets before moving into the next part of my talk. So India is the second largest producer of millets next to China, but majority of the produce goes into preparing animal and bird feeds. In area-wise, Rajasthan and Maharashtra tops. However, in terms of production, you can see that Uttarakhand, Tamil Nadu, and Arunachal Pradesh leads the race. As far as TN is concerned, evidences in literature even indicates that people were consuming millets before rice and wheat came into the picture. Out of my curiosity, I have searched the poems of Sangam literature that was instituted, 5,000 years ago. And interestingly, I found that foxtail millet and kodo millet were predominantly used in agriculture and trade. If you could see that there are two main literatures like Purananuru and Agananuru, and there are several marks, and there are several places where these millet, foxtail and kodo were repeatedly cited. It seems that even for the god, they have mixed blood with the red foxtail millet and they offered, as this song from Agananuru indicates. And a song mentions that it is mentioning the features of Mullai Nilam. It shows that at this place, rice cannot be grown, where it is only suitable for foxtail and kodo millet. Coincidentally, a paper published last but the before month traced the origin and spread of rice, and the report shows that Oryza sativa indica, what we consume today, had a hypothetical ancestral origin from this region in the northern India around 4,000 years ago, and it spread across the other parts of Asia and as well as southern India. So this came only 4,000 years ago, where millets were there, before that. So slowly rice took the place of millets. One interesting thing which I want to tell you, like here, is there is one QTL that is in rice that is called Southbound QTL. It was present, now they have introgressed into several varieties, but previously it was present only in the varieties cultivated in Odisha and Sri Lanka, which also underlined kind of fact that migration of people from Odisha to Sri Lanka, who later became the present day Sinhalese. So coming back to the story of millets, I'm showing you the composition of protein, fat, ash, crude fiber, carbohydrate, etc., etc. You can see that millets are far better than the cultivated rice, wheat, and oats. In addition to this, millets are also rich in bioactive compounds, and to know more, as well as it is also rich in micronutrients including potassium, calcium, phosphorus, magnesium, sulfur, zinc, iron, boron, manganese, and nickel. Millets are provenly superior than major cereals, and in addition, millets are also rich in bioactive compounds, as I said. And to know more, one can refer to my paper that is published in 2016. So yeah, I'll spend 2 minutes to show you what something unfortunate has happened. But what is even after having such a wonderful, nutritious crop with us, our people are much inclined towards consuming pseudo cereals like oats. Primarily, oats was cultivated as a feed for cattle, and the European nations are the largest contributors to oat production. If you could see, oat is not cultivated in India, and we import predominantly from the European nations and few parts of Australia and America. And oat cannot be consumed by humans as such due to digestibility issues. When these nations overproduced oats, or when these nations produced oats in surplus, they devised a nice plan to make it edible for humans and sell it in countries like India. If you could see the nutritional info given in any oat pack, you can see the term called rolled oats. It means that the oats is physically and chemically processed at a large scale to make oat flour or bran or flakes, which is edible. We Indians have this tendency of buying and eating whatever that is advertised on TV. So we are largely feeding on this processed horse food, and we are thereby contributing to the economy of some other country. So I am moving to the next section by leaving a question to you all that goes in line with our Prime Minister's recent statement of going vocal about local. Why we people are more into consuming these imported oats when we have our own indigenous millets, just because they are not, getting advertised on the television. So yeah, but on the other hand, millets are also gaining popularity, but slowly. As you could see, a restaurant in Delhi is offering Banyard millet biryani. In Madurai, I heard from a TNAU professor that the farmers are shifting to millet cultivation from rice. You could see that in Kotambati, it is a region near Madurai, yield goes very high for millet farmers. So yeah, millets are slowly getting importance without any such advertisements on televisions. On the other hand, recognizing the nutritional potential of millets, our central government has declared all the millets as nutritional nutri cereals in the year 2018. And we also recommended the United Nations to declare the year 2023 as International Year of Millets, which has been formally accepted. We will be celebrating the year 2023 as the International Year of Millets. Our Ministry of Agriculture wrote to FAO, and it was formally accepted. Yeah. So now coming back to the question of, you may ask, okay, you are talking about millets, but you previously showed about hunger. Now, how we are going to bridge this gap? How we are going to bridge the gap between the hunger hotspot and the accessibility to healthy food? The answer is given in one of my papers. I have worked out this, where I mapped the 55 hunger hotspots reported by United Nations and integrated the data of millets marginally produced in those hotspots. FO means foxtail millet, PR means proso millet, LI means little millet. Likewise, we have mapped the millets those are consumed in such hunger hotspots. Interestingly, one or the other millet is being cultivated in predominant hotspots, but might be in less quantity or by a small community. So one durable way of solving the hunger at these hotspots is to promote millet cultivation as well as consumption that requires a coordinated effort of international body like UN as well as the local government. One simple pipeline which we have proposed is, it involves 5 steps. That first thing is, we have to identify the crop that is being grown in those hunger hotspots, or a crop that is suitable for cultivation in such hotspot. Second is to devise a plan for cropping. This includes working on irrigation, pest pathogen control, soil management, weeding, etc. Third is to implement a plan to promote this scale into a large scale production to reap the maximum benefit out of it. Fourth is to improve the crop management program and also to involve biotechnology to improve the varieties being cultivated. Fifth is, or this should go hand in hand. And fifth is the storage and supply. Post-harvest storage is more important to stock up the food grains to meet food demands during abnormal situations. And if the supply chain gets disrupted, actually people will be having some reserve to consume. So this also helps in strengthening food security, alleviating poverty, and increasing the resilience and sustainability of farming systems. Though this flowchart looks very simple, it requires tremendous efforts. It should include, or it should involve different agencies and public to achieve a measurable success. That also we should be, making a note. And this has to be done at a quicker phase, because as of now we are thinking that we have only 2 problems: COVID-19 and the economic recession which is going to follow this COVID-19. But actual thing which we are forgetting is the big impact that is very bigger than the ongoing pandemic. So that is what is called as the climate change. It is climate change that is going to have a huge impact on food and nutritional security of the global people. So that is where I'm coming to the second part of my today's talk of how to use technology to improve the crops to produce better varieties. So before going into the subject, one thing we have to understand is how these crops came, how the crops which we cultivate today came. One is the evolution, which happened for millions of years. If you could see, rice, millet, sorghum and maize, they all diverged from a common ancestor about 48 million years ago. Before that, it was a single crop. At 48 million years ago, they got diverged into different crop species. So this is how evolution has happened, but evolution happened in a very slow phase. On the other hand, our ancient humans, they started domesticating. So domestication is again very slow. If you could see the wild and the domesticated crop, you can see the difference. When you select for particular trait, it takes years and years to achieve such traits. You could see the wild Zea maize or the wild Zea species, it looks like this. And the cultivated Zea maize, the present day cultivated Zea maize is this. Oryza, wild rufipogon. Wild is Oryza rufipogon. Cultivated is Oryza sativa. This is the wild potato and this is the cultivated potato. This is the wild tomato where the fruits are berry shaped. This is the cultivated tomato. One very good example of domestication, or one very complex crop domestication example, is the domestication of wheat, which run for years and years. If you could see wheat, the present day wheat, we got it out of several hybridization, whole genome duplication and chromosomal shuffling events that happened between different species and within the genomes also. So it happened, thousands and thousands of years to reach the present day wheat. So this is how this domestication has happened. Whatever we are consuming today has been domesticated from the wild. That one point you have to understand. Third is breeding. Whatever the variety which we are cultivating today, then better varieties which we call as by our present day farmers, those were released through breeding. But the breeding process itself is slow. One very good classical example of breeding is rice variety IR8. Many people would be remembering of this IR8 and IR64 for sure. IR8, if you could see, the program started in 1962 by IRI, International Rice Research Institute in Philippines. And in India, it came in 1967. So it took several years to reach India. Even if you could see, last year, a variety called Super Nigiri 1 was released to the farmers. Yeah, is my screen visible now? Artha, is my screen
yes.
Okay.
So you can proceed. Yes.
Okay. Yeah, I'm just going behind 2 more slides to bring back the continuity. So what I was saying is, we have identified several genes in our own lab that when we overexpressed in rice, it is conferring tolerance to different stresses, particularly to drought, and mode of action of one such gene I have explained. And I'm not going into this. Yeah. Another gene which I was showing you is this ASR4, which was isolated from again millet. I'm showing only millets as specific examples to demonstrate to you people that such an underutilized crop has so much of genetic information which can be useful or which can be directly used for the genetic improvement of cultivated crop species like rice, wheat or maize. So when they overexpressed this SIASR4 in Arabidopsis, this is the overexpression line and this is the wild type line. When they stopped watering it for 15 days, still the overexpression lines were able to survive. And when then they were watered from the 15th day for the 3 days continuously, the plants are back to normal where the wild type gave up. Same thing they did it in rice. This is the overexpression line, this is a silence line, and this is the control. So they stopped watering for 10 days, and after 10th day, they started watering for 3 days, and if you could see, the rice totally recovered. Another gene is this LTP, SILTP, Setaria italica lipid transfer protein. When they overexpressed in tobacco, when they stopped watering for 10 days and even up to 21 days, the plants were able to recover. So it shows that if the plant has this gene, and if there is no water for more than half a month, the plants will be able to recover whenever there is water. So such a wonder, if you could see, these genes can do in plants. The same thing they did it in rice. So these are overexpression lines, and this is control. If you could see, drought was given for 10 days, and after 10th day, our control plants are recovering. So this is for 22 days, and this is for 28 days. So then we thought, now we have to identify all those genes which can do such, which can confer such tolerance traits in crop plants. So we identified, or we standardized a pipeline for identifying potential genes and gene families, and we have published several papers on each of the gene families and we kept this resource open for the general public or the millet research community to use the candidate genes identified in these papers for overexpression in rice or any other crop. This is a very recent one published 2 months ago, where we have studied the C4 photosynthetic genes. So yeah, not just overexpression confers good traits. Even sometimes if you knock out or knock down a gene, then also you will achieve some better traits. Knockdown could be done through RNA interference or virus-induced gene silencing, where knockout present day we are doing it using CRISPR-Cas9. So I will give you a snapshot of what is being done in millets alone, because my talk today should be focusing on the underutilized crops. So in millet, they have shown that Setaria viridis is the wild cultivar of foxtail millet, and they have shown that by silencing a single gene called Ph.D using RNAi, they say that the cell wall ferulation and biomass digestibility gets modulated. So it increases the biomass digestibility. So if you could silence a single gene called Ph.D in Setaria viridis, it increases the biomass digestibility, so that the straw of these plants can be easily used for feed for the fodder. It can be fed to the cattle. Coming to virus-induced gene silencing, it is a very new thing for millets specifically, although it has been largely worked out in several crops, even in plant species like tomato, Arabidopsis, Nicotiana, soybean, rice, wheat, sorghum, zea mays, etc. For millets, as I said, it has received very less research attention so far. So whatever that has happened in major crops, the same thing will take several years to happen in millets. So if you could see, for virus-induced gene silencing, a vector has been identified in 2016, but till date no report is there on the use of virus-induced gene silencing to silence any gene in millets. So yeah, how it works, I'm sure that many people might be knowing, how virus-induced gene silencing works. That is if you want to target a gene in the plant genome, say for example, I'm going to target this particular gene in the genome, what I will do, I'll clone a part of this gene in the viral genome. So automatically when the virus infects the plants, it inserts its genetic content into the plant, and it uses the host machinery to synthesize the complementary strand. When there is a double stranded DNA, or DCL and DRB recognizes it and it cleaves into small interfering RNAs, or what we call as siRNAs. These siRNAs could then bind to the mRNA of the target gene, and that activates this risk agoproteins to cleave the mRNA. So if the mRNA is cleaved, and there won't be any translation. So this is how WIGS works. So, yeah, as I said, for weeks in millets, several, protocols have been established, but so far, no study has reported the use of these weeks to silence any particular gene for to achieve any agronomic trait or any stress tolerance or any such notable trait which will be useful for the farmers. On the other hand, we now have CRISPR, although CRISPR has been extremely or widely used in several crop species. The use of CRISPR in millets are very limited. Again, the reason is the same, because these crops are less as invited. These crops have invited less research attention. So CRISPR is something relatively new to foxtail millet. I assume that there would be several first-time learners here. So first, let me tell you or give you a brief introduction of how this CRISPR works, and then we will see how this has been effectually used in other crop species to improve traits. First thing is CRISPR means CRISPR-Cas9 means this Cas9 complex. Cas9 complex has a Cas9 protein with a guide RNA inside it. This guide RNA, the sequence of the guide RNA will be complementary to the sequence of the gene which we are targeting. If I am targeting a particular gene, what I will do, I'll design the guide RNA having the complementary sequence of my target gene. And when this Cas9 goes and binds to that gene, so this is the gene, what it will do, the Cas9 has two nucleus domains. These nucleus tend to cut the DNA. So the DNA will be cut, and when the DNA is cut, there happens this non-homologous end joining repair, which introduces some random bases to join the DNA. So when these random bases are inserted, there could be a frame shift, or there could be a missense, or that could be a nonsense, or that could be any aberrations that stops the actual function of this gene. So by this way, the gene can be silenced. By this way, the functional gene can be totally stopped. So in foxtail millet or in wild millet, Cas9 has been now established. Although it has not been published, it is just in a preprint server. But now they have devised a Cas9 system for multiplex editing. Multiplex means you can edit more than one gene using a single vector. You can see that, for example, within a single vector, you can have several, target guide RNA sequences, so that it can edit several genes in a single go. So gRNA1, gRNA2, like that it is given, right? So this is what is called as multiplexed gene editing. One example, or one very good example, I will show you of how CRISPR-Cas has helped in reducing the issue of farmers. If you are from a state where wheat is cultivated predominantly, you might be knowing that this pre-harvest sprouting is a big issue in wheat cultivation. A farmer, when he cultivates the wheat and when he stores it, the wheat starts sprouting. If it happens, then this becomes totally useless. So it causes a tremendous yield loss for farmers. So just to stop this, several scientists have attempted several ways with different approaches: overexpression, knockout, knock-in, knockdown, etc., but nothing gave a promising result. When CRISPR came into picture, one group, this Abe et al., what they did, they silenced a gene called QSD1. If you could see this GGG of this QSD1 gene has been edited to different letters, ATT. Wheat has three genomes, right? A genome, B genome, and D genome, because it is hexaploid. It is a hexaploid. So just by changing GGG into ATT, the pre-harvest sprouting has been arrested in wheat. So this is what a genome editing approach can do to a crop plant. Just by changing a few letters, you will be able to achieve a tremendous or a drastic change or a drastic positive phenotype in any crop that is being cultivated. Not just for trait improvement, we can use CRISPR-Cas9. You can see that this is a wild tomato that is called Solanum pimpinellifolium. It has an indeterminate growth. Those fruits are very small, of pea size, but it is relatively stress resistant. What they did, this group in 2018, they targeted just 6 genes. Gene 1, Gene 2, Gene 3, 4, 5, 6. They targeted 6 genes and they edited these 6 genes in the wild tomato. And what happened is the wild became cultivated. It had determinate growth. It has large and more fruits. It had 500% high lycopene content and it is stress resistant. This is Solanum pimpinellifolium domesticated. So de novo domestication, like I have shown you, right? I mean, in the beginning of the second part of the talk, the domestication of wheat, it took years and years, but de novo domestication using CRISPR-Cas9 can happen in 8 to 15 years. So you can introgress or you can bring or you can mutate some negative genes to make a durable or a strong strong domesticated crop within a short span of time. So this is the potential of CRISPR-Cas9. And now our Department of Biotechnology, Ministry of Science and Technology has issued a policy note where they are going to now approve or where they are going to now agree to the release of genome edited organisms. This is available in the website because they have faced lots of issues with the genetic engineering, BT crops and so on. So this time they don't want to take a chance, it seems. So they have released, or they are already in favor of releasing genome edited species. So they have already released the regulatory framework and guidelines, and very soon it will come into effect. And if that case come into effect, then there might be chances that a researcher can release a genome edited crop for the betterment of farmers. So yeah, as I said, as of now, agriculture is not focusing on these two issues. Agriculture is largely focusing on the bigger issue, which is actually in place here, and it is going to get better or get bigger from now on, because these two affects us all, but this one affects specifically the farmers. So when the farmer gets affected, the crops get affected. If the crops get affected, the food security gets affected. If the food security gets affected, then everybody gets affected. So if there is no food because of no farmer or no crop, then the life and livelihood of everybody is questioned. So we are now majorly focusing on addressing this food security or this thing using different interventions. That is where I have shown you how overexpression OE of different genes helps in promoting or in increasing the climate resilience of crops, where knockout helps in modifying or in knockout and knockdown in improving the yield contributing agronomic traits. So that is what the importance of biotechnology is. So why this is important and why I'm always showing this climate change is that it is very near, as I said, and from the inland areas you could see that dryness is getting spread. And one fact which you have to understand is we have only 2% of the world's land, but 40% of fresh water. When fresh water is only, 4%, sorry, not 40%. When we have only 4% of world's fresh water, on the other hand, we have 16% of world's population because we are the second most populated country. So we have to have sufficient water for the population and we have to have sufficient water for the crops. So we have one single source of, several limited source of fresh water, but we have many beneficiaries in both in terms of humans, cattle and crops. So water is getting reduced. If you could see from the inland, the dryness is going to spread across different states, whereas the coastal areas are kind of secured, but the inland areas, the dryness is getting intense. So inland areas are at the higher risk of economic losses due to rising temperatures than coastal and hilly areas. Crop failure, I have shown you how bad the locust attack was. Now you know that there is no rain. We have late monsoon, heavy monsoon, that is what I call as erroneous rainfall. Pests and insect attacks are higher, diseases are higher, and other natural calamities. And always what happens is, like, if drought is there, then some other infection will come. So there will be a combined stress. A plant will be facing several stresses at a given point of time. So a small cropland, when it has to face different stresses, from outside the environment, heat will be there. From the land, drought will be there. Sometimes a disease will follow. So at a given point of time, if a crop is given 3 stresses, the crop will lose focus. And now it is unlike animals or humans, it cannot move. It has to stay at the same place where it has been planted. That's what we call as sessile. Plants are sessile. So when they cannot move, they have to face the challenge from the place where they are planted. And when the stresses are intense, they have to give up. So plants die, but you should note that plants will not die easily. Plant will fight back as much as it can. Only when it realizes that it's not possible to fight the stresses, it will give up. So crop failure due to climate change is very high. Locust attack, I've shown you how it is bad. And FAO is saying that drought accounts for 83% of yield loss in agriculture, followed by other factors. So drought is the main culprit. That's why I've shown 3 examples of overexpression of foxtail millet genes in rice, tobacco, and Arabidopsis, which helps in circumventing this drought stress. And farmer suicide, you might be reading in newspapers then and there, which shows that our farmers are committing suicide because of crop loss, debts, and other factors. It's not just crop farmers, it is also the workers who works in such farms because of loss of employment. So after 2016, the cumulative state-wise data is not available from any of the websites. But 2016 data, you can see that farmers are dying because of several climate change related issues. And as it is being said that when all the farmers are gone, it would be our turn. So our turn is yet to come. So this is one hand, malnutrition, undernutrition, and obesity. Those things I have talked in very detail, and I also shown that our South Asia, including India and its neighboring countries, are very much prevalent, or we have a higher proportion of such undernourished or malnourished children under the age of 5. Although we don't have the exact numbers on our hand, but we could see that the condition is pretty bad in our country. And this will continue. This is what FAO is predicting. FAO is saying that by 2015, the crop yield will decrease by 25% if we don't address climate change now. Now the question is how to address climate change. The answer is addressing climate change is not as easy as it is being told on paper. Ice or glaciers has already started melting in polar regions. And one climate report which was released by our Honorable Health Minister, who is also the Minister of Science and Technology, Dr. Harshvardhan, he released an assessment of climatic conditions in India, and and that data shows that our own Himalayan ice is getting melted because of high temperature or rise in temperature there. So climate change, we are seeing it for real. We are visualizing loss of rain. We are visualizing erroneous rainfall. We are visualizing insect pest attacks. We are visualizing locust attack. We are visualizing different diseases that are coming into the plants. So addressing climate change is not an easy task, but making a crop resistant to the climate change is a relatively easy task. practicability and practicality are two important things which I am considering before making this statement. So addressing a climate change is not a quick task, but making the crop resistant to such climatic deviations is easy. So that is where biotechnology comes into picture. That is where I have shown you manipulating the gene or genome using different approaches. Let it be overexpression, let it be knockout, let it be knocked down, let it be knock in, let it be anything that involves wigs, RNAi, CRISPR, such things which I have shown you or which I have given you a glimpse of helps. So that is where we repeatedly underline that biotech should be acknowledged by the public and the policymakers, and release of such varieties shouldn't be hurdled because of unscientific reasons, and such things has to be made at the policy level to facilitate the release of several varieties. Several varieties have been made, but we were not able to release it to the farmers just because of policy issues and loss of public awareness. So those things has to be removed. That can secure not only food and nutrition security, it will also secure the life and livelihood of farmers, and it will also address the hunger and malnutrition issues which is prevalent in South Asia, as I have shown you in the world map pictures, which is also very much prevalent in a country like India. So that is what is the take-home message I have for you people. I finish my talk by telling you another Tirukural which says that if a human has to beg and live, the Tirukural curses the creator of such human that he should also beg and perish. So by this, I wind up my talk, and it is over to Partha and his colleagues, and I can address if you people have any questions. Thank you.
Thank you so much, sir. Sir, your presentation is quite interesting and so informative. And those literatures taken from Tamil is really authenticates your work on millets. Your knowledge fascinates the emerging technology of genome editing technology, the field of genome editing technology. And the lecture was so informative. Sir, thank you so much. And shall I open session for question and answer session? There are questions from our participants. Let me read out the questions to you, sir.
Yes.
The first question is from Nivedita of our participants. Sir, isn't knockout better than knockdown, as it will possibly completely eliminate the unwanted gene? Why not focus on knockout methods?
Yeah, when it depends upon the purpose. If we are experimenting on a gene in lab, we don't want to totally eradicate the gene. In that case, what we do, we used to silence it. So in that case, we go for knockdown, then knocking it out. So it depends upon where you want to use, what is the end use of that approach. So we cannot judge which is better, but we can use it accordingly, or we can use it according to our purpose of use. That is the difference.
Okay, sir. So it's a participant from Gopinath. In upcoming generation, any possibility to getting naturally cultivating products like rice, tomatoes, etc.? Why? Because nowadays the people using chemicals during cultivation period.
Actually, he is indirectly meaning like, why shouldn't we go back to this organic farming and
yes.
thing? That is what is the actual essence of the question.
Yes.
So again, like we people have already dumped tons and tons of chemicals in the field, and we have already made the soil to lose its fertility. So fertile soil is not there as much. So it needs some supplements to enhance the growth of the plant. At least the farmer should not go into loss by cultivating or going such in an organic way, because once those who have those who have cattle in their home, they might be remembering, or they might be knowing that there used to be a pit in the home itself, where they dump the excreta of the cattle there, and when it gets filled, they will take it to the field and they will put it on the field. So those things are not happening nowadays. So we have altogether moved to a different phase where we are now totally dependent on such chemical fertilizers and synthetic elements. So coming back to the natural thing which he is asking is a bit different task. Given that we are now far away from what we were 20, 30 years ago. Even before Green Revolution, we were doing this organic farming only. Green Revolution brought so much of chemicals and fertilizers in a incentive. What they call is giving incentives. So such things brought these chemicals into the picture. Now eradicating the chemicals and going back to nature is a difficult task. It is a difficult task actually.
Yes. Sir, another question from Geetanjali. Non-specific lipid transfer protein is also found in finger millet, but it is associated with alpha amylase inhibitor activity. Whether the both are orthologs or they are different genes.
Okay. They belong to the same gene family, but I don't know whether the genes are the exact orthologs. That has to be studied. Yeah.
Another question from Preeta Chakrabarti. Is genetically modified organisms like BT cotton, golden rice, roundy soybean are related in this topic?
Can you repeat the question?
Genetically modified organisms like BT cotton, golden rices, soya beans are related to this topic or not?
Yes, it is. I'm advocating for those, to be frank.
Yes. Some more questions are here. Millets takes longer to digest and liberates lesser energy than rice. Phenolic components in millets also interferes with thyroxine to decrease metabolic rates. Can consuming them on alternative days help?
Okay. There are several questions in that single paragraph. So first thing is, what is the first part of the question? First sentence.
Millets takes longer to digest and liberates lesser energy than rice.
Okay. When you take rice, when you consume rice, within 20 minutes of consumption, your blood glucose level will shoot up. That is because rice has much of what is that called as rapidly digestible starch. So immediately the glucose will be released into the bloodstream. That is why rice we are not recommending to diabetics. Coming to the millets, millets are rich in a kind of starch that is called as resistant starch, RS. These resistant starch cannot be digested in the stomach or in the small intestine. Rather, when it goes into the large intestine, it gets digested by the gut microbiota. So those gut microbiota acts on the resistant starch, it catabolizes them and releases